Hello students, good evening you all. Welcome to another live session from Brilliant Education. In this class, in this video session, we are going to discuss one of the complicated chapter, biotechnology. So far, we have been discussed different chapters like molecular basis completed, principles of inheritance also completed. But if you look at the weightage of these two twin chapters, according to CBSE sample paper, for this two biotechnology twin chapter, 12 marks given student. So all of you with joy sit in front of YouTube or what they call laptop or tab, whatever it may be, and all of you, I'm requesting all of you to take a NCERT with you. NCERT textbook. I will show you some reference also from the NCERT. All of you open the NCERT and sit. Am I clear? And in this chapter, first there are two chapters are there, biotechnology principle and biotechnology application. Out of these two chapters, first we will start with the process. What exactly the construction of rDNA, what are the steps are there, gel electrophoresis, PBr322, PCR method, bioreactor method, lot of complicated terms are going to come. And today you have to ask me a particular doubt, you have to say the answer, you have to ask me a particular, sir, this topic and that topic. Today most of the question you are going to do, I will be silence of most of the question. You have to answer the questions. Keep in mind all of you. Shall we move on to the first question? Right. Look at the first question. This question from NCERT example. First question, a little tricky question. So look at the question, what the question says. A bacterial cell was transformed with a recombinant DNA. A bacterial cell transformed with a recombinant DNA molecule that was generated using human gene. Human gene we inserted into the bacteria host. Fine, done. Now look at here. However, the transformed cell did not produce the desired protein. The transformed cell, whatever the bacterial transformed cell is there, they did not produce the human protein. What is the reason? Reason could be, okay, look at here. I am going to cover two more questions also in this MCQ. So carefully listen here what I am explaining. The first, human gene may have intron which bacteria cannot process. Amino acid codons for human and bacteria are different. Human protein is formed but degraded by bacteria. All of the above. My dear students, if you look at the option 2, according to genetic code, it is a universal means what? The codons which are code for the amino acid is similar in human, in bacteria, in all type of organism except mitochondrial DNA plus some protozoans are given. So second option is not possible. I make clear? It is possible when the construction of or getting a human insulin or human proteins in bacteria, because the genetic code is universal. So here this is not correct answer. B, leave it. And C, human proteins are formed but degraded by the bacteria. It's not given in the question. It's bacteria not produce that protein. Look at the option 1. Human gene may have intron. Come on, all of you go back to the molecular basis once again. If you go back to molecular base of inheritance, splicing we discuss, eukaryotic genome, eukaryotic genes have intron exon. So splicing takes place there. But what about the bacteria? If you able to recall biotechnology application chapter student, Eli Lilly company has started the first artificial human insulin production, right? So which DNA they inserted into the bacteria? cDNA. Let me take one second. Okay, look at here. First you take the gene. Look at here. This is the human insulin gene. You have to first let this gene undergo transcription. transcription, right? After that, what will form? mRNA. This will form mRNA, and after that you have to allow this mRNA to what? Tell me one more. Reverse transcription. Remember, all of you, reverse transcription to form double-stranded DNA, we call it cDNA. This cDNA you have to tag with what? Plasmid. You have to tag with the plasmid to form rDNA, because if directly human genome you insert it into the bacteria, our desirable protein will not come, because our DNA, our gene has intron, exon. You have to process that one artificially, and after that only the bacteria can synthesis. You got my point? You understand my point? Right. So the right answer of this question is option A. How many of you guessed this answer? How many of you got this answer? Right now. It's good. Hiba, correct. Now look at the next question. I'm going to the next question. Next question. Look at here. This is a well-known question. What is the critation of, so for the DNA fragments movement on agarose gel during gel electrophoresis? What exactly the critation? Any one of you? Pavitra, Raina, Ala, Ganga, any one of you? There is a small critation is there, the movement of a DNA particle from one to another, that means the negative to positive. Don't know. Come on, any one of you answer. Right, very good student. Ganesh, correct. The smaller the fragment size, the further move, right? So option A, the positively charged fragment move positively, DNA is negatively charged, right? Easily we can remember D, deoxyribose, N stands for, here it is nucleus, but we can remember negative. Negatively acidic in molecule. So DNA negatively charged. So negatively chargeable molecule DNA, if it is a smaller molecule, it will be go fast and further move towards what? Positive charge. So our answer is option A. Remember, all of you, when you are doing the biotechnology chapter, gel electrophoresis process, the principle of gel electrophoresis will learn separation of DNA fragments based on the size. Remember, all of you, gel electrophoresis question every time, diagrammatic questions, or direct they're asking illusion topic question, or they're asking which chemical you're using to visualize that DNA particle. Lot of questions are coming from gel electrophoresis. So before you complete the biotechnology, gel electrophoresis topic, thoroughly prepare for the final examination. Look at the next question. Next question. This is very easy and very important question, also key, or the tools of biotechnology, key tools of biotechnology. If you wanted to do a rDNA technology, you wanted to construct one new rDNA. So what are the weapons? What are the tools you need for the rDNA technology? The first one, which of the following contains the key tools for recombinant DNA technology? Restriction endonuclease enzyme. Yes, restriction endonuclease enzymes are very important. How many of you tell me, how many of you are going to tell me the discovery year of restriction endonuclease enzyme? Discover year of restriction endonuclease enzyme. Albert, yes. Khalid, which year? Devananda, Abin, which year restriction endonuclease? Very no, Allah, notes not correct. 72 first rDNA constructed, but before the first rDNA constructed, there must be a endonuclease enzyme has to discover, right? 63, absolutely. Who is this? Ganga, very good. 1963 restriction endonuclease enzyme discovered in bacteria. Remember, wait a moment, hold a minute, everyone keep in mind, restriction endonuclease enzymes are the exclusively secret or come from where? Found in where? Bacteria. Restriction endonuclease enzymes are defense mechanism of bacteria. Restriction endonuclease enzyme so far 900 from different type of bacteria, dash different type of bacteria. From dash different type of bacteria, 900 restriction endonuclease enzymes so far have been identified. Can you fulfill this, filling the blank? Any one of you? Absolutely, get 230 different bacteria. Am I clear? Right. So restriction endonuclease enzyme, important weapon. Ligase enzyme also important weapon, important tool. Vector also. Actually, there are 5 key tools. Remember, it can be asked for 2 mark question, student. 2 mark question also they can have 5 tools. One is called restriction endonuclease enzyme, ligase enzyme, vector, host, polymerase enzymes. If this 5 without without any one of these 5, you cannot construct the rDNA. Recombinant DNA, for the construction you need this 5, but the host little late. For the construction of rDNA, first 4 are important. After that, the rDNA you have to transform to where? Host, remember. So I can write what first one is correct. Ligase enzyme correct. Host organism also correct. Polymerase enzyme also correct. Vector, Taq polymerase enzyme, and primers. Primers are not a part of, right? It's not a part of key tools, right? So leave it. What are primers? Primers are single-stranded RNA molecule. That I will tell you little later when question comes. Next, restriction endonuclease enzyme, ligase enzyme, and bioreactor. Bioreactor not a tool of key tool of biotechnology. Leave it. So the right answer we can tell, right? The first one and also second one. So option B. Am I correct? Am I clear? Right. Very good. Next question. Next question we are going. The restriction enzymes are used in genetic engineering because. Why? Why this restriction endonuclease enzyme we are using in the biotechnology or genetic engineering? What exactly the purpose? We have discussed this one. These are molecular scissors. These are the one which can cut the DNA at a specific position, right? Look at the answer. They cut the DNA at specific base sequence. Okay, appropriate answer is there. There the nucleases can cut the DNA at the variable sites. That sites the palindromic sites or the specific site. Am I correct? Albert, yes. Sana, good, very good. So the right answer we can write what? Option, okay, Minha, good. So they can degrade the harmful protein. How can they degrade the harmful protein? No, beta. So just think, read the option one more time. 100% good. You should not lose that. You should not lose at least one question, beta. One question answer in where? Final examination. 60 out of 16 out of 16 you should score. MCQ. You should not leave. You should not wrong answer that one. Am I clear? Now we can write what? They can join different. Joining by ligase. Joining by ligase enzyme. So leave it. So answer is option A. Am I correct? Tom, Ganesh, Khalid, yes, very good. Next question belongs to exclusively online student. I will take rest. I will take rest. You have to do, you have to read, you have to answer. Am I correct? Am I clear? Next. 2 bacteria found to be very useful in genetic engineering. I don't know the 2 bacteria. You have to find out the 2 bacteria. Which 2 bacteria are genetical engineering useful bacteria? Khalid, Minha, Allah. So what is the answer? Who is this? Right, good, very good. Jade, Kitraj, good, very good. So the right answer is Nitrobacter, Azotobacter, Azotobacter, Azospirillum. These 2 bacteria we have seen someplace, right? We have seen someplace Azotobacter, Azospirillum. We have seen in microbes in human welfare. Exactly, yes, microbes in human welfare. So answer is what, beta? Echerichia and also Agarobacterium. What about the second? Rhizobium. And here it is Azotobacter, Azospirillum. Sir, what kind of bacteria? Can any one of you guess? Any one of you guess? Azotobacter, Azospirillum, free-living nitrogen fixation bacteria. Am I correct? Free-living nitrogen fixation, right? So here Echerichia coli we can use as a host, right? This is the host. Agarobacterium tumefacium can use as a vector, plasmid vector. Any one of you answer me? Agarobacterium tumefacium can cause disease in dicot plant. What is the disease called? So particular disease is caused by the Agarobacterium. Dicot plant NCERT is giving us. What is the disease called? Any one of you? Any one of you? Yes, absolutely. Crown gall disease. Remember, crown gall, tumor making, cancer making disease. Agarobacterium. But we modified that bacteria according to our convenience. Remember. Next question. So answer is option D. Next question. Can you do this simple question? Simple question. I have fully confident on my student, they will do this now. Any one of you? MTK, Allah and Kitraj, Tom, Angeline Jacob, Abin, Hiba, Raina. Yes. Now answer me here. Recombinant DNA is achieved by the cleaving the DNA. Pro DNA is cleaving, cutting, cleavage. So restriction endonuclease, simple answer, restriction endonuclease. Restriction endonuclease enzyme, 2 mark question. I am going to cover 4 points. Everyone should remember until NEET exam complete, not your CBSE. Until your NEET exam complete, this 4 point on your tongue. Number 1, restriction endonuclease enzymes exclusively found in bacteria. It's a defense mechanism. Restriction endonuclease enzyme can cut both DNA strands. Restriction endonuclease enzyme cannot cut on single stranded DNA and single stranded RNA. Restriction endonuclease enzyme, each restriction endonuclease enzyme we have a 4 terms. What are they? Genus name, species name, straining, and also based on discovery the number. And it also inspect the length of DNA. It cut the DNA at a specific site we call palindromic site, little away from the palindromic site. These all points everyone should remember for final exam. Next question. Very simple. I love match the following, student, whatever you say. I love the match the following, because match the following is the one which give a hint to us, right? Allah, Sana, they give hint to us. No need to think. Just they will give a hint. We can easily answer that one. Very simple questions. I am also requesting to the CBSE to make all 16 MCQs are what? Match the followings only. Right. Now look at here. Bacillus thuringiensis. So what is the answer? Bacillus thuringiensis BT. Bacillus thuringiensis BT what? Bacillus thuringiensis BT. Cry protein. So I am writing that cry protein directly, right? Cry protein. Next one is Thermus aquaticus. So DNA polymerase. DNA polymerase, other name here. The particular polymerase enzyme coming from the Taq polymerase. Thermus aquaticus, the Taq polymerase, right? Taq polymerase, right? Now, Agarobacterium cloning vector. So far 4, 3, 1 Salmonella 2. 4, 3, 1, 2. So I am writing right 4, 3, 1, 2 option B. Am I correct? Option B is correct, right? Simple answer. Taq polymerase. Can any one of you tell me Taq polymerase enzyme can work under dash temperature at? Taq polymerase enzyme can work also. They did not denature. They work under at exactly 100 degrees. That's a thermoacidophils. These bacteria are thermoacidophils. Remember. Next, high temperature. The linkage of antibiotic resistant. The linkage of antibiotic resistant gene with the plasmid vector become possible with. Read the question carefully. They are asking linkage. I will cross check. Ganesh, C. Okay, good, very good. So I was expecting you will give that restriction endonuclease, exonuclease enzyme or endonuclease enzyme. But the question asking what linking two DNA particle joints. When two DNA particles are joined, the enzyme name is called ligase enzyme. So DNA ligase. If this type of question, carefully remember, you have to mention also after this two DNA fragments cut with the restriction endonuclease enzyme, you have to add the ligase enzyme to join these two particles. Remember. Next question, question number 9. Very simple question, and you have to 2 more question also I will cover, but you can also learn 2 more question from here. There are 4 different type of target cells are there. For example, it is a bacterial cell. If it is a bacterial cell, I can use the enzyme called what? Tell me. Lysozyme enzyme. If it is animal cell, protease enzyme. If it is a plant cell, I can use cellulase enzyme. If it is a fungal cell, fungi cell, chitinase enzyme. But here, look at here, they are given alga, which is mismatch. They are given mismatch, correctly match for the organisms well, and which one of not correctly matched? Not correctly matched, mismatched one. So which one is mismatched? Alga. Here it is cellulose or pectinase. So methylase. So this is the wrong answer. Fungi, chitinase, bacteria, lysozyme. Human health and disease. If you are able to recall, Hiba, do you remember this? Next, Khalid, in human health and disease, lysozyme enzyme present in our saliva. It is a physiological barrier. Tears and also saliva, physiological. How many of you remember this? Do you remember this? MTK and Hadia. Do you remember this? This is a physiological barrier which can kill the antibiotic in nature. Look at here on my finger, thousands of bacteria, billions of bacteria might present. If I keep in my eye, nothing will happen, no infection, because in my tear there is a lysozyme enzyme which act on bacteria. This is our physiological immune system, innate immune system. Until and unless bacteria concentration very high, I will not get infection, because in my tears and my saliva, lysozyme enzyme is there. So we can dissolve bacterial cell. Answer is option A. My clear? Next question. Question number 10. Exclusively for you people. This topic 3 mark or 5 mark, very, very important. Selectable markers to identify your recombinant or transformant and eliminate the non-recombinants and non-transformer. 2 types of selectable markers are there. One is called antibiotic resistant gene we can use, and second one is laxy gene. Carefully read and answer the one. Answer this question. Any one of you. Hadia, can you do this? MTK, can you do this? Hitraj, Hiba, look at here. Colonies of recombinant bacteria appear white color. Wait here. Let me do this one. Okay. Here, what I'm doing. This is plasmid. I separated from bacteria. This is bacteria. From this bacteria, I separated what I'm separated? Plasmid I separate. Plasmid have laxy gene. This is laxy gene. Done. In between the laxy gene, I inserted my target gene. This is my target gene. I inserted here. Exactly here. I inserted here. Am I clear? If this plasmid receive my DNA, it must that laxin must have what? Inactivated, right? So this is my recombinant. Now if I send this one here into the bacteria, I am keeping this bacteria in ex-gal or synthetic lactose. This synthetic lactose, in presence of beta-galactosidase enzyme, it white color convert into blue color. But if it is remain white color, what does it mean? It means our insertion, our DNA has been gone in the inserted in the target place. That means in the laxy gene. So laxy gene must have inactivated. Based on this equation, based on this principle, chalo find out the answer. The colonies of recombinant bacteria appear white. That means our gene has gone. Our gene has inserted. In contrast to blue colonies, if it is blue colony means what? Non-recombinant, of non-recombinant bacteria. Because of, I am going to read your comments. What is the right answer? Devnanda. What is the right answer? Samuel. Can you read the? Can you answer me? Can you answer this question? Insertional inactivation of beta-galactosidase in recombinant bacteria. Absolutely, this is appropriate, absolutely correct. Come to the rest of 3. What's wrong in the rest of 3? Inactivation of glycosidase enzyme. We did not discuss this one, leave it. Non-recombinant bacteria containing beta-galactosidase. Beta-galactosidase enzyme is there in where? Recombinant one, non-recombinant one. Beta-galactosidase enzyme still function in where? Non-recombinant, what they are asking. But this is not correct. But the colonies are converted into white color because insertional inactivation of alpha-galactosidase in it's not recombinant bacteria. It is a recombinant. That means your gene, your inserted gene, whatever the specific gene you inserted has gone. It is called insertional inactivation. So the colony, that ex-gal synthetic chromogenic substrate convert in it still remain white in color. All colorless is given in the NCERT. If it is remain colorless, our gene has gone. So we can select that what they call bacteria, or you can select that transformer as your recombinants. If that chromogenic substrate which convert into blue color, beta-galactosidase enzyme still working, that means your gene did not go. That means your inserted gene did not go. So insertional inactivation, carefully prepare from the NCERT. So I can go option A. Question number 11. Simple. You might have all of you are done in your school practical examination. This one, I think so. So what is the right answer? Is this one? Adding a chill ethanol to a DNA and extracting the DNA called. Any one of you? Right, spooling process. So DNA spooling. This is a simple. Next question. Carefully read this question and find out the answer. Carefully read. The different steps involving in the process of recombinant DNA. They are given, different steps is given and they are randomly given. Different steps are given and the different steps are randomly given. 1, 2, 3, 4. They are not given the sequence in order. Find out the rest. Now here. Now arrange them in correct order. Correct order they are asking. We know of one important step of our DNA. Isolation of DNA is the first step. Isolation of DNA is the first step. So where is isolation of DNA? 3. First. Okay. Now here it is. I am writing 3. After that, you have to amplify the DNA. You have to amplify that particular gene. So 3 leads to 2. After that, you have to ligate the DNA with fragment of vector. So I am writing what with what? 4. So when you are ligating your DNA with vector, that is a step inserted in the construction of RDNA. Right? So I can write this one is RDNA construction. Done. Next one is, after this one we can write what? Insertion of recombinant into host. Yes. Into host cell, we can say what this is called a transformation. RDNA you have to transfer to host cell. The host could maybe bacteria, could maybe plant cell, could maybe animal cell, fungal cell, whatever. You have to use the host to get what? Product. So what is the last step here? Why we are sending into the host? To get a large amount of product, right? Do you remember the bioreactor steps? In the bioreactor step, what we are doing? We are increasing the number of bacteria and finally we are getting the largest scale of product, 100 liters to 1000 liters of product we are getting. So I can write last one is called what? Tell me, extraction of the desirable gene product. So 3, 2, 4, 5, 1, 3, 2, 4, 5, 1, option B. Remember all of you, I am requesting all of you, RDNA technology steps. There are 2 diagram is given in CRT. One is the stepwise explanation of RDNA. You have to draw the diagram. It can come for 5 marks. Go and prepare that what they call a sample papers or previous paper. RDNA technology 5 mark question. One is a diagrammatic representation and one is step by step representation is there. Right? Answer is what? Option 2. Which of the following step are catalyzed by Taq polymerase in the PCR? Polymerase chain reaction has 3 steps: denaturation, annealing, extension. A question: who discovered PCR first? Albert, Kitra, anyone? Who discovered Shadi? Who got that Nobel Prize? And who got what the first discovery of this PCR? Any one of you? Okay, the right answer is C. Fine. So what is my answer? What's the answer of my question? What I'm asking? PCR first developed by, first explained by. Very good. Who is this? Allah. Absolutely. Carey and Millers. Remember all of you, this type of question can come. They ask Carey and Miller first time they discuss so they describe what they call the process of what? PCR. So PCR Taq polymerase in PCR, extension of primer end on the template DNA strand. I have one more question. In PCR, how many primers we will add? How many primers we will add? 3, 4, 5, 2. We add 2 primers, right? 2 primers to opposite of what? Parent DNA strand. Remember, primers also they can ask. Next question. Following statement describe the characteristic of enzyme restriction endonuclease. Identify the incorrect statement. Identify incorrect statement. I'm reading your comments. Come on. Any one of you? Sana, Shadi, Adin, Hadia, pick up the right answer of this one. This is very important. Any one of you? No. You should not see the textbook. You should think only. Who is that? Ganga and Albert. Any one of you? Right? Good. Very good. Hiba. The enzyme binds to DNA specific site and cuts only one of the two DNA strands. I told you, I clearly informed you, I clearly mentioned 4 important steps of restriction endonuclease. It will not act on single-stranded DNA and single-stranded RNA, and it cuts both DNA strands. It cut both DNA strands. It could form a blunt end or sticky end, different type of end based on the type of restriction endonuclease enzyme. Next, option A. Now, next I am going to the next question, question number 15. This is a big question, but if you know a clear information of Agrobacterium, you can easily answer this question. Very easily you can answer this question. Which of the following statement incorrect? Number 1, DNA being a hydrophilic molecule cannot pass through cell membrane. Absolutely correct. Wait a moment. Before I go to the second option, 1 minute. Do you remember competent host making topic? Competent host making. To increase the permeability of host, we are adding something. We are adding something. Any one of you tell me? We are adding something to the host to increase the permeability to what? DNA. Competent host. Think. Very good. Calcium ions. Absolutely. Calcium ion. So that calcium ion can increase the so in that topic, clearly it's given what? DNA, hydrophilic molecule, cannot pass through. So it cannot easily go. Bacterial also will not accept. No, you keep us outside. I will not allow you. Now here what happened? Agrobacterium tumefacium delivers a piece of DNA known as zDNA. Oh my God. You know, student, there are 5 types of DNA. How many of you know? Watson and Crick explain dash type of DNA model. Watson and Crick explain dash DNA type of model. Dash type of DNA model. Any one of you? A, B, C, D, Z. B DNA model. Absolutely correct. B DNA. According to B DNA, in one turn, dash base pairs. In one turn, 10 base pairs. Yes or no? So this is a zDNA. Remember all of you. A DNA, B DNA, C DNA. Let me write here. A DNA, B DNA, C DNA, D DNA are right-handed. But zDNA is a left-handed DNA. Agrobacterium tumefacium have a tie DNA, tie plasmid, tumor-inducing plasmid. So this is a one small hint to pick up. Now what incorrect statement? Which transfer normal plant cell into tumor cell as directed this tumor produce chemical against the correct? Retroviruses, so this statement is wrong. Retroviruses, adenovirus, papilloma virus are also known to use a what? Cloning vector in animal. If you able to recall, option C is close to gene therapy, right? In gene therapy, ADA enzyme deficiency therapy, we use what? Retrovector. So this statement is correct. In genetic engineering, DNA from different sources are cut within the same restriction endonuclease enzyme. Yes, your target DNA plus vector DNA you have to cut with the same restriction endonuclease enzyme, because the ends will be different, because the end will be same. If you use the different enzyme, and the ends will be different ends. So different ends cannot what they call bind. Remember all of you. So answer is incorrect statement is it is not zDNA, sir. It is tie plasmid or tie DNA, or I can write directly tumor tDNA. I can write tDNA. Next question. Good. Very good. Very good. Absolutely correct. Next question. Primers are. Carefully read what are primers. In case of the DNA replication process, PCR process, we have learned primer. Molecular base of inheritance, we have learned primers. Remember all of you, small chemical synthesized oligonucleotide, it is maybe 10 to 18 or maybe 6 to 20 also we can tell. Small short nucleotide. Which nucleotide? RNA nucleotide. So I am writing here, it is RNA nucleotide. nucleotides, RNA nucleotides of about 10 to 18 nucleotides that are complementary to the region of template DNA. My dear students, this is NEET level question. Everyone I'm requesting, look at the paragraph, look at the diagram of PCR. In the PCR, primers are added to the opposite of third prime of template strand. Look at there. Look at the diagram. Two primers added opposite to the third prime of parent DNA strand. So it added to what? Opposite. Why, sir? Why the primers are adding to the opposite of third prime? Any one of you can guess? Any one of you can guess why we are adding the primers opposite of third prime? Sir, new DNA can synthesize from. I will be stopped. Come on, any one of you? Joel, next Jade, Shadi. New DNA strand synthesis from 5 prime towards the third prime. You got my point? That's why we are adding the primers to opposite of third prime. So our answer is option A. Primers are complementary. Chemically synthesized oligonucleotide about 10 to 18 nucleotide that cannot complement. If it is not complementary, why would we add? Why would we add? So leave it this one. Next, the double-stranded DNA that need of amplifying. Next, specific sequence of presence of recombinant DNA. The appropriate answer A. It is a 10 to 18 nucleotide complement to the template strand. Next one. This question, carefully look at here. There are vectorless gene transfers are there. Biolistic gene gun method, electrophoresis method, chemical method. I'm requesting you all, Hiba, mark this one. Shadi, Allah, everyone mark this one. Hafiz, in the exam if question come, what is the chemical which is help the DNA to insert into the host? That is polyethyl glycol. This polyethylene glycol is a chemical method which used to send gene transfer without vector. So it is called a chemical method. Remember all of you, without vector gene transfer methods are 4. Number 1, chemical method called polyethylene glycol method. Number 2, electrophoresis method, micro injection method. Third one is called, any one of you, biolistic gene gun method. Remember this 4. In this 4 one, we can expect 2 mark question also. This 4 method. So carefully before your preparation. Read better. Next, a selectable marker is used to. Simple selectable marker. So I selected. I am genetic engineering now for one genetical engineering faculty. I am student of genetical engineering. I am doing to construct a rDNA. Okay, I cut the DNA, target DNA with restriction endonuclease enzyme. I cut that vector also, but I select the vector which don't have a selectable marker. Every vector must possess ORI. We discussed this one. ROP, selectable marker. It should be recognition site. It should be small in size, right? Small in size. Now in size. Look at here what it is given. Selectable markers are This selectable markers is helping you to pick up which vector receive your DNA. That means what is recombinant, what is non-recombinant, easily you can pick up. So we can write helps in eliminating the non-recombinant. If it is not received, we can eliminate. So why should I take this when you did not receive my DNA? Why would I take you? Why would I culture you? It's a waste of my money, waste of the enzyme, waste of time, right? So I have to eliminate them. I have to eliminate that vector which did not receive our gene. So how can I eliminate this one? I can eliminate with what? Selectable marker. Identify the gene with the desirable. No, we cannot identify the gel electrophoresis. Select the suitable vector for the transformation into a specific crop. Mark a gene on the chromosome for the isolation. These selectable marks are not for this all. Simple identification of recombinants and eliminating non-recombinants. Remember. Next question. Very simple question. Pick up the right answer. Now here, if a recombinant DNA carrying antibiotic resistant gene, that is ampicillin, is transformed into E. coli cell, the host cell is transformed into a ampicillin resistant cell. The ampicillin resistant, oh my god, this much of big question is given. The answer is very simple. Any one of you? Devananda, what's the right answer of this one? Yes, absolutely. Adin, good, very good. Any one of you other students? Now, all of you, Shadi, Adin, next, Albert, Allah, right. The simple answer is option C. Option C. Are you with me? Are you with me? Am I clear till here? Everything clear? Right, good. Right, good, Angeline, right, correct. Especially. Next question. The recognition site. This type of question, answer me. My dear children, I am clearly telling you, it's been long period. I am shouting in the class also. Do you remember? PBR322 diagram. PBR322 diagram, very, very important. Artificial vector. They are asking BAM H1 restriction endonuclease enzyme cutting site where it is present. If you don't know the entire PBR322 diagram, it is very tough to pick up the right answer. The recognition site of BAM H1 in PBR322. I am not reading the option. Directly I am coming to the comments. I have confidence on my students, so I am going to read the comments. Good, Angeline. Good, Hiba. Sana, Hadia. Nice, very good. So this is tetracycline resistant. Very good. Absolutely. Can you please, any one of you, tell me ampicillin resistant site, how many endonuclease restriction sites are there? What are they? In ampicillin place, how many are there? In ampicillin site, how many are there? If I am not wrong, Sal, PVU, PST, I think so. Yeah, PVU, PST, right? Yes, absolutely correct, Ganga, absolutely right. This two, even in the BAM H1, or even in the tetracycline also two. Even in the tetracycline, in what they call in tetracycline resistant also two. One is BAM H1, second one, any one of you? One is BAM H1, second one, Salve, absolutely, Tom, absolutely, very good, it's Salve1 also, very good. Next question. The treatment of host cell with bivalent or divalent cation leads to the... I will be silenced. No, I am not biology teacher, I am a commerce teacher. No, I don't know this answer. You have to help me out from this very dangerous condition. Any one of you? Tom, right, good. Is it correct? Is it correct, Ganesh? Yes. Now, the treatment of a host cell with divalent cation, just now we discussed calcium ions, fine. So the answer is maybe C or D. Who is that? Kidraj, yes. Look at increase the efficiency with which the DNA entered into the absolutely. Option B. And we have to treat this one with what? A little temperature. Heat shock method, we have already discussed this one. Heat and shock treatment. Remember. Now, next question. Next question is little important and you have to tell the answer. I will be silenced. I will wait here. Incestual inactivation, you know. So tell me the answer. Incestual inactivation mechanism we have already learned. Answer me. Incestual inactivation of the recombinant DNA is inserted within the coding sequence of... I need answer. Who is that? Allah, good, very good. Khalid, Ganesh. Beta galactosidase. Option A, beta galactosidase. I am going to molecular base of inheritance. Okay, tell me, beta galactosidase enzyme in Escherichia coli turn off when? It turn off, it will not undergo mRNA beta galactosidase enzyme when inducer absence or inducer present. Who will going to stop this mechanism? Any one of you? Any one of inducer absent? Khalid, absolutely correct. Inducer absent, somebody is going to interrupt this one. Who is that? Who is that fellow is interrupting this one? Any one of you? Any one of you? Absence, okay, fine, correct. Somebody is interrupting the expression of LAG-Z gene. Absolutely. Who is this? Very good, Hiba. Absolutely. Repressor protein. Repressor protein go and bind to the operator and not allowing the RNA polymer. Absolutely. Very good. Next question. Now look at this. I told you PBR 322, no doubt. PBR 322, this type of question can come. A, B, C, randomly they will give and they ask you to pick up the find out that correct one, which endonuclease enzyme, which tetracycline, this selectable marker they ask. Look at here. Identify A, what is A, B and C. On the top of this CLA and HIND D3 there is eco R1 also there, right? Eco R1 also here there is a eco R1. Eco R1 is there. Devananda, is it correct? Eco R1 is Ganesh. Yes. B just now discussed here. B along with Sal1, BAM H1. BAM H1 restriction site. And what is C? Don't look at the option. Tell me what is C. What is C? C. What is C? Any one of you? Good, very good, very good, very good. O-R-I, absolutely correct. I have a question, one oral question, 1 minute. Look at here. A, B, C, out of these three, one is essential for make your desirable DNA into many copies. Many copies. Your desirable DNA into many copies. Adin, Angeline, Sana, Samuel, I need answer. Yes, absolutely. ORI is the one. NCERT clearly mentioning that one, the ORI site can responsible for making of thousands of copies. So our answer is option. I can go option A. Eco R1, BAM H1. C. What is that? ORI. What is D here? What is D here? Ampicillin resistant. Just now I told you in ampicillin resistant two sites are there. What are they? PVU1, PST1. This diagram very important, my dear student. If the same question or same diagrammatic question repeat in final examination, you have to remember Sandeep sir, okay? You have to remember everyone, I'm telling you. Everyone, whoever listening, Angeline, Allah, Kitraj, you have to remember my name. Sandeep sir is saying in that YouTube live class in Brilliant. Okay, beta, remember. Next question. Which of the following is not future of the plasmid? Not future of the plasmid. No. I don't know. You have to pick up. Not future of the plasmid. One thing. It is a not characteristic feature. All characteristic feature you have to select. It is circular or not, ORI is there or not, selectable marker is there or not. But one thing is not there. What is that? Yes, absolutely. What is that? The one thing which is single stranded. If it is single stranded, your desirable DNA is double stranded. How come double stranded DNA like it with single stranded? Common sense, right? Think, right? So answer is option C. Next question. Which of the following options are not correct regarding eco R1 enzyme? Before you go to this answer, any one of you tell me eco R1 make what type of ends, and after that answer this question. Eco R1 make what type of ends? Just I need what type of ends? Flip flop, come back, welcome back. Tell me what type of ends it form? Yes, sticky ends, absolutely. When sticky ends are formed, there is an unpaired base pairs are there, right? There are two unpaired base pairs are there. That means eco R1 cutting something. Let me write here. So palindromic sequence 5 prime is G A A T T C, and this is third prime. Fine. What about this one? Third prime C T T A A G, and this is what? 5 prime. So eco R1 exactly cutting here. So that means between the two nucleotide, which bond is there? Phosphodiester bond is there. So if you cut this DNA, carefully look at here. It cut the phosphodiester and hydrogen bonds also cutting. Look at here. After that, it making the two unequal size of DNA particles, DNA molecules. So if you read the options, one place you are stuck in, I think so. If you read the answer, absolutely, oh my god, most of the students are correct. Restriction endonuclease enzyme, fine. And isolated from ECO RY13, absolutely correct. Cut a specific position within the DNA called palindromic, yes, palindrome sequence, fine. It cuts only hydrogen, no, so phosphodiester bonds also. So phosphodiester bond plus hydrogen bond. So we can write what? The not correct one is option D. Am I correct? Good, very good. Next question. Next question. These questions are very important, and the theorists, whoever attend my theory can do, but for the other students also I will explain. Can you do this answer? How many fragments will be generated in a linear DNA? This is a linear DNA, eukaryotic linear DNA this is. So I am treating this linear DNA with restriction endonuclease enzyme. How many restriction endonuclease enzyme? Molecule digested with the restriction enzyme having four recognition sites. Let me take here now 1, 2, 3, and here it is right 4 places. How many fragments are forming? Fragment number 1, fragment number 2, fragment 3, fragment 4, fragment 5. Remember everyone, if a question come how many fragment they are asking, but you have to read the question. It is a linear DNA or circular DNA. If it is a linear DNA, number of endonuclease enzyme plus 1. How many here? 4. How many fragments will form? 4 plus 1, 5. So answer is option B. Am I correct? Absolutely. If it is a circular DNA, if it is circular DNA, okay, let me take it is a circular DNA. This is circular DNA. Okay, how many fragments here? So how many is the enzyme? 1, 2, 3, 4. Fragments 1, fragment 2, fragment 3, fragment 4. So number of restriction endonuclease enzyme equal to the number of fragments. In case of where? Circular DNA, remember. So next question I will be silent. You have to pick up the right answer. I don't know the right answer of this question. Now, what about this one? Come on, flip flop, do this better. And who is this? Allah. How many fragments will be generated? A close. Look at here. Circular. Ganga, come on, do this. DNA molecule digested using the restriction endonuclease. How many? 6 restriction site. 6 restriction site. Yes, absolutely. Thank God. Good, very good. Who is this? Right, absolutely. You have to remember this carefully. In the question they are giving circular or linear. Linear in case of us, our eukaryotic is linear. Circular means bacteria, prokaryote, remember. So answer is what? Option C. Option B. Okay, I can write like this. Option B, 6 fragments. Question, next question. Which of the statement given is incorrect statement? Incorrect statement. In micro-injection method, foreign DNA is directly injected into a nucleus of animal cell used by the microneedle. They are asking incorrect statement. Statement 1 is correct or not? Appropriate look like a statement is correct, sir. Okay, fine. Keep aside. Micro-injection method is used in oocyte. Yes, egg and embryo. Right, done. Gene transfer directly into the animal cell. Electrophoresis. Electrophoresis. Do you remember Salmonella typhi bacteria perforation in small intestine we discussed? Infection in the small intestine, perforation, small hole formation. Perforation means hole formation. Same thing in biotechnology application chapter. Cry protein activated under alkaline medium and make a poration in the gut of insect. We discuss poration means hole formation. Look at here. Electroporation in the formation of temporary pores in the plasma membrane of host cell by using lysozyme or calcium chloride. Is this make sense? Students, carefully read the electrophoresis method. We are treating the bacterial cell with a very less voltage of current. So the small holes are forming with the whole DNA entered. So I can write incorrect statement is C. So what's wrong in D? D is good or not? Look at the D. In chemical mediated gene transfer method, certain chemicals such as calcium phosphate helps in the DNA. Yes, this we already discussed, bivalent cations. Remember, yes, absolutely done. Good. Next question. PCR method. Carefully look at the question, read the question, look at the diagram and find out the answer. Hiba, Sana, next. Any one of you. Lots of biology. Nice name, beta. Chalo, answer this one. The given figure below shows ABCD of polymerase chain reaction. Select the option giving correct identification. So 3 steps is given. Do you know PCR? Polymerase chain reaction, 3 steps are the annealing, denaturation, annealing, extension. Pick up the right one. B, denaturation. Where is B? B is here. B is not denaturation. Okay, let me read. At the temperature of 98, no sir, denaturation takes place is 92 to 94 degrees Celsius. Fine. Keep aside wrong. A, denaturation at temperature of 50 degrees Celsius. No, it is no. A is denaturation, but it is not at 50 degrees. C, extension of presence of heat stable DNA polymerase. What is C? This one is C. A, annealing with 2 sets of primers. I am going to read your comments. Yes, absolutely. Oh my God. Good. Very good. Absolutely. So that means I can understand you peoples are perfect in biotechnology. PCR perfect. So answer is what? We can write option C. Good. Very good. C is extension. Taq polymerase enzyme we are doing. Along in the extension step, along with the primer, something also we are adding. Any one of you can guess what is that something also we are adding? Along with the Taq polymerase enzyme, we should also add along with the Taq polymerase enzyme, we should also add along with the Taq polymerase, we should also add primers already in the annealing. Thymus aquaticus, fine. Along with that one, Ganga, along with that one, something we have to add. No. Oh, exactly. Who is that? Sana. Absolutely. DNTPs we should add. We should add free nucleotides. Otherwise, how can it elongate? How can it will be elongate? We have to add the free nucleotide, right? DNTP. Yes, exactly. So the answer is what option? See, next question. Which of the following is not required for the preparation of rDNA, recombinant DNA molecule? Not required. I am not talking about that. It is not required for the getting a genetic engineering product. rDNA construction. For the construction of this recombinant DNA, you don't need something out of this 4. Any one of you? Flip flop. Lots of where are you? Answer me. Good, that's correct. DNTP correct. Now answer is E. coli. Absolutely, very good. E. coli cell is no need. Good, very good. Restriction endonuclease. Yes, otherwise you cannot cut the DNA to your specific area. Yes, we need ligase also. We need for the ligation. DNA fragments also we need. E. coli, when we will need this one host? After construction of rDNA, for the transformation to get a product, we need the host, right? So for the rDNA construction is not required. The answer is option D. Question number 31. Which of the following is commonly used for the vector for introducing DNA fragment in human lymphocyte? A, D, A, gene deficiency. We discussed this is in gene therapy. So any one of you, therapy, any one of you. Come on, flip flop. Very good. Absolutely good. Correct. Retrovirus. Retrovirus genetic material. What is the genetic material of retrovirus? What is the genetic material of retrovirus? What is the genetic material of retrovirus? Single stranded RNA, ssRNA. Human health and disease, we discussed this one is a HIV virus, retro family. Stirred tank of bioreactor have been designed for. for the getting of a huge product, we have to culture that transformants in the bioreactor, right? So in that bioreactor, we are providing everything, pH, temperature, food material, everything we are providing. But one thing we are continuously giving, what is that? Yes, absolutely correct. Availability of oxygen throughout the process, NCERT line direct it's given. Availability of, because aerobic bacteria, if you are stopping this one, it will die. So we have to increase the number of bacteria. If more bacteria are increasing in the bioreactor, more product we can separate, we can get. In bioreactor, culture of exponential growth of bacteria. Remember, next question, 33. I have 35 questions, 3 more questions. Don't leave until I finish 35. Okay, stay with me. Which of the following bacteria is not source of restriction endonuclease enzyme? Not source. It is a not source of restriction endonuclease enzyme. Anyone, not source. So number 1, Haemophilus influenzae, HA, what is that? HAND, HIND2, HIND1, these all are from Haemophilus influenza only. Escherichia coli, EcoR1, EcoR5, EcoR4, good, very good. Next, Agarobacterium tumefacium, no restriction endonuclease. It is a big modified vector to transfer the gene in plant cells. So answer option C, Bacillus amyloides. Here BAM H1, BAM H1. BAM stands for bacteria name. Next question, which of the following not component of downstream processing? Not component of downstream processing. Any one of you? Not a component. Flip-flop, you should not give me a question mark. You should tell the right answer. Approximately all the answers you are giving correct only. No, remove the question mark. You have to give a full confidence with answers with full confidence. Keep in mind. So which of the following component of downstream process? Separation is there? Yes. Purification is there? Yes. Preservation also. Expression already done in bioreactor. Expression already done in bioreactor. So leave it this one. Am I clear? Right, done. Very good. Next question. 35, last question, assertion reason. I will be silent. Really, I will be silent. You have to pick up the right answer. Albert, Kedraj, Sana, Shadi, Yusuf, lots of biology. I need answer. Assertion, the tumor-inducing plasmid, type plasmid of Agarobacterium tumefacium act as a cloning vector in recombinant DNA technology. Correct. It's true. Right. Done. The type plasmid which is used in the mechanism of delivering gene to cell remain pathogenic. Open the NCERT. Look at the line, what is given. The line is given. Agarobacterium bacteria now modified into deliver the gene into the cell without making any disease, no more pathogenic. Very good, absolutely. So answer I can write what? Assertion correct and reason is false, option C. Right, so this is our biotechnology process chapter. Thank you very much for attending brilliant classes. All of you, next class we are going to discuss human health and disease or biotechnology application. Any one of these 2 chapters I will discuss. Do not miss and share this video to your friends who absent today and whoever close to you. Am I clear? And like this video and share this video to your friends. Don't miss my next class. Not my all brilliant classes. Don't miss any brilliant classes. Thank you. Good night all of you. Thank you.
Brilliant Qatar, your trusted coaching partner for IIT, JE, NEET, Science and Commerce tuitions with 10 years of excellence in quality training. Brilliant Qatar.